Okay, so thanks for inviting me here. Today, the sort of main thing I'd like to get to is work that's collaborative with these people shown here. Cameron is a postdoc in my group. Salvador is a graduate student, and then my colleagues Krishna at Northwestern and Arvind at University of Chicago. So in order to get to this main point, I'm going to have to start with some background. So let's go back about 4 billion years. There's a lot of talk for the past decades about this being the information age and the information revolution happening, but biology did that a long time ago. One could, I think, easily argue that the origin of life was an information revolution when chemistry somehow started encoding information in polymers or other kinds of molecules, and then using that information to dictate other kinds of chemical processes. That kick-started evolution, and now we have systems, living systems, that are really information-based chemistry. That's sort of the core of what living things do. So if we accept in very vague terms that everything is code of some sort, the question is, what kind of code is it? How do we think about information in chemistry in a way that takes that sort of metaphor to something actionable? And for that, we need some kind of models or concepts for how information is stored in a molecular system and how that information is used. In other words, molecular algorithms. So what are the natural models for biomolecular algorithms? So if we look at the kinds of information systems, computing systems that we know today, we sort of have two prototypes, electronic computers and brains. And we have classes of models, digital circuits, programming languages, the Turing machine, sort of kicking off the theory of computation. And now we have neural network type models that try to incorporate ideas about how brains work. So is one or the other of these really the right way of thinking about information-based chemistry, say at the molecular level? I think I would argue actually that both of them can be. It depends upon the circumstance. But the real goal of at least this talk is to sort of ask, how do we look for biomolecular algorithms? How do we recognize them? When will they be under our noses, but we can't see them? So for that, we have to know where to look. If you talk to physicists, one kind of answer you might get is that living systems are out of equilibrium. They have a metabolism, they take energy, they produce heat and waste. And so are the computer systems we build. They generate a lot of heat. Brains take an inordinate amount of energy as part of our metabolism. And so there's maybe a tendency to think computation and algorithms in living systems will be the non-equilibrium processes, and that's where you should look. There is, however, some caveats you might consider. Quite a long time ago, Charles Bennett asked the question of how much energy does it take to compute, and he showed that you can basically take any algorithm, non-equilibrium algorithm if you with, and convert it to a reversible physical process that can be driven forward with arbitrarily little energy. Maybe it's slower, but it still goes. And the trade-offs, in some sense, aren't ridiculously bad. There's another sort of line of reasoning that suggests maybe non-equilibrium is, in some circumstances, a bit of a red herring, which is the success of neural computation models that basically treat the computation as a random walk on an energy landscape. And these are very old ideas also.
What do you mean by arbitrarily low energetic cost?
If you say you have a reversible Turing machine, its state space can be thought of as a one-dimensional line. This is a very special case. It's easy to analyze. And if it's a random walk, you can go forwards or backwards because it's reversible. You can put an arbitrarily small bias so that forwards is just a little bit faster than reverse. The difference between the forward and reverse rates is a measure of how much energy per step you're using. And that random walk will drift forward with an arbitrarily...
So it might take arbitrarily long time.
It is slower, yes. Okay. Yes, like, typically...
End of the universe, okay.
No, it's not that bad.
Charlie did his work back before stochastic thermodynamics. So there are things called the thermodynamic speed limit theorems that apply also to CTMCs, which wasn't even there when he investigated things. Moreover, the models that he looked at, there was no actual complete cycle where you then reinitialize for a new run of the computer. So there are massive subtleties. I would highly recommend look up Naoto Shiraishi. He's got a great physiograph letters on the thermodynamic speed limit theorems. They, at a minimum, demonstrate that Charlie's claim is not actually right if you want to do things in finite time. This is complicated.
You know this much better than I do. I acknowledge that. Nonetheless, there's something there. And I do believe that those results tell us a little bit about where we can look and what we can think about. And there's certainly inspirations for me for what follows, and maybe you'll have some objections to what follows as well, but let's get there and see how bad it is. Okay, so I titled this talk, Equilibrium is Inference. This is sort of my thesis. There are two problems with this thesis. One is that it's not true, and the other is that it's not new. So equilibrium and inference are distinct concepts, yes. But what I'm hoping is really this is an exhortation rather than a scientific claim. It's that when you're thinking about things that think, things that infer, things that work with information, don't too quickly go past equilibrium models because there might be something interesting there. I tried to look up this quote for this talk about equilibrium versus non-equilibrium. It's attributed to Ulam and von Neumann, both of them, of course, sometimes about equilibrium, sometimes about nonlinear, but the basic point is, there's so much that is nonlinear. Are you really, or so much that's non-equilibrium, are you really sort of confining your study very well? That's one sort of caution one might have. But the other one I think that I'm trying to say is that elephants are actually quite interesting and quite powerful, so think about them first before you get too into the other things. So what can you see there? So for equilibrium, in this talk at least, I'm going to be talking about a very basic statistical mechanical point of view. You have some reachable state space. You have some energy on your states. You have some neighbors. There's moves between state X and state Y and back and forth. Detailed balance is that the rates of going forwards and backwards are related to the energies of those states. And given that, you will equilibrate to the Boltzmann distribution, which is a nice analytical form. So lovely tractable. Inference is, again, you have a state space, you have some a priori probability. Now you get some information. Maybe that information is that you're in some particular subspace of events, y, and your posteriori probability, probability of x given y, your probability of x is now changed now that you have this information, and that's the conditional probability distribution. You might, there's different kinds of information you can think about. You might have a subset of variables. For example, maybe your state space has, that's a vector, and you have some visible units and some hidden units. If you know what the visible units are, then you have a posterior probability on the hidden units. It's the same thing. Just to visualize that briefly, there's some true joint distribution. You have marginals summing the columns, or a marginal summing the rows. If you have some information that says something about what y is, then you're in a confined subspace, and you get your conditional probability. So we can call that, computing this is the process of doing inference on the information that you've received. Okay, we'll also talk about learning in a little bit, and the big picture there is there's some true distribution. You have a model that is parameterized such that it represents the wrong distribution. You get some samples, you update your parameters, and ultimately, hopefully, your model will start looking like the true distribution. Okay, so in these low-dimensional spaces, this is fairly straightforward. What becomes interesting is when you look at very high-dimensional models, and as I mentioned before, energy-based neural network models are a nice sort of framework, classical framework. Just to remind you, the Hopfield associative memory was sort of one of the touchstones for people thinking about energy with respect to neural network models. You have some kind of update that's doing a linear weighted sum, maybe with a bias, and asynchronously choosing an element. You can turn it on if that's a positive sum, otherwise you turn it off. And you can learn weights by Hebbian learning on a set of examples alpha. And this system sort of respects an energy function, which is just a quadratic sum plus a linear sum for the biases, such that every update will decrease your energy if you change state. So that means you can envision the computation is this process of dropping a ball at some initial conditions, and then it rolls down to the bottom of some well, and the state there is your memory. So here are some examples of that, where you start in a state that is maybe a very noisy instance of one of the memorized examples, and as you update, your energy goes down, and eventually you settle into a local minimum. That's the final recall. So this model has an energy, but it's not an equilibrium model. You get trapped in a local minimum, not necessarily the deepest minimum. There's no probability that respects the energy here. But that changed with Sejnowski, Ackley, and Hinton when the Boltzmann machine was defined. We still have a fully connected system, but we now maybe have some visible units and some hidden units. The hidden units give it extra power. But we still have exactly the same energy function. It's just now some of these variables are hidden units and not just visible units. And we have a stochastic update that respects detailed balance such that the system will equilibrate to a Boltzmann distribution. And one of the things that's valuable about this is that we have a new sort of simple, still simple learning rule, this contrastive learning, or some people call it wake-sleep approach, where during a wake phase you have Hebbian enhancement of weights, and during a sleep phase you have an anti-Hebbian sort of disruption of correlations. And this essentially sort of fills in holes in the energy landscape when you're sleeping, and this digs holes so that when you are no longer changing, your correlations are matched between the wake-sleep phase and the sleep phase. What's nice about this is that you no longer think about the energy landscape as representing these sort of local pits, but you can store you can have the system represent larger parts of the state space, and in fact can have continuous attractors like a ring attractor, where the system maybe drifts around in these low energy states, which in high dimensions can be quite interesting. So Boltzmann machines can represent much more interesting probability distributions. So one of the classic examples of probability distributions, MNIST. We would like to sort of sample this distribution of handwritten digits that look like digits, but aren't sort of specifically one of a finite set of specific digits. So this is just an example of a high-dimensional representation. OK, I've already introduced the Boltzmann machine. Although it's fully connected in what follows and what preceded a little bit, I'll just represent a specific example where we have a 10 by 10 array of sort of the images for a reduced resolution MNIST, class nodes. This is another subset of the units, which are saying which class is it. This is sort of an inconsistent state that thinks it's class 1, 5, 7, and 8 all at the same time, and some hidden units that are helping the computation. And so you have a weight matrix that basically encodes for a distribution, and as the system runs, it's exploring that distribution, and the states are wandering around. Great. If you can't see this very well, if you just look at a sort of temporal window time average, you can see that it's really sampling these digits. And if we look at the energy of the states, you can see it's just sort of uniformly fairly low, bouncing around. You can think of that as a ball that's sort of bouncing around near the bottom of a continuous attractor, and it's moving around the attractor from the 4 to the 5 to the 9 to the whatever else as it moves along this continuous attractor, which might be higher dimensional than shown here. But this is what we mean by generatively exploring a probability distribution. OK. Inference. Inference in systems like this correspond to clamping. So if you're given some information, one way of sort of making the network respect that is you just set certain units to the values that you know, and you let the other ones continue to do their random walk. So here we've clamped everything sort of in orange. We've clamped the image, and now we get as the system explores, we sample a probability distribution of the hidden units and the class nodes conditioned on the input being 7. OK, and it does its random walk. We could also sort of do it the other way. We can clamp the class nodes and let the digit nodes and the hidden units do whatever they want. And again, we can see that as it gets to lower energy states, it starts to look like a 7. So it's inferred, based on the information about the class, what the digits should be in a probabilistic sense. You can, of course, put information in different ways. You could say the bottom half of the image is like this, and it's not a 9. So what's it going to be? It thinks it's going to be a 7 in this case. So how do you train a system like this? You've got these hidden units. You've got some visible units. And if the system has a set of weights, then there's some probability that the visible units will do something. That's the marginal, because you don't care what the hidden units are doing. You're just looking at what the visible unit is doing. And then you have your data set, which is describing the target distribution over visible units. And the classical Boltzmann machine learning is looking at the relative entropy, the distance between what you want the system to do and what it does do. You take the derivative, do a little bit of algebra, and you get that you just, the change with respect to this particular weight is the correlation when the system is free minus the correlation when it's clamped. So that gives you a gradient descent learning rule. OK, so you clamp the machine to sample this. You run the machine and sample this. It's not a very efficient learning rule, which is why no one uses it. But it has a nice physical basis, and it can deal with a fully connected network. So being able to deal with a fully connected network is powerful if you were trying to look at biological things that maybe won't respect the restrictions that make it easy to compute. Your biological network is everybody is talking to everybody else somehow. And so you want to understand how systems like that can be thought of as doing inference and how you can train them. OK, so this is an example of the learning. You sample when you take a sample from Q, you clamp it, the hidden units do whatever they want to do, and that's these correlations. You let it run free, that's those correlations. And you'll be taking different samples to collect your statistics for the clamped distribution. The interesting thing is that this learning rule doesn't distinguish between hidden units and visible units. That's, I think, the really fascinating thing about it. OK, so that's the background. I'm now about to launch into sort of the new stuff. But I'll take a little intermission about technology and science. We all know that technology can drive science. One of the classical examples is talking about elephants. That was the name of this early stage steam engine. You start building things. Somebody starts thinking about how they work, develops some mathematics, and then that mathematics gets generalized into other kinds of contexts. So thermodynamics sort of developed as the theory of the steam engine. And now the concepts that were developed here, entropy, enthalpy, all the rest of it, has become a language that we can talk about things as distinct as, say, protein folding, right? So you can't really talk about protein folding without understanding thermodynamics now. So when we think about what was good about this technology, it's not just that building the technology gives us trains that allow, say, a molecular biologist to order and transport samples from one state to another state so that they can do their experiment. That's great that you have that power, but more importantly, it gave these concepts that we can use to understand biology from a new perspective and a rigorous one. And so if we think about the technology of building computing systems, and the theories that derived from that, both Turing machine-like things and modern computation, the value of computation to science isn't just that you have technologies that can do things for you, can give you answers about analyzing systems, but also they introduce concepts for understanding what biology is. So to make that step, let's remember that the Hopfield model was sort of initially conceived of as a generalization of Ising models, which was actually a model of magnetism, where there's an energy that depends upon whether neighboring cells are the same sign or opposite sign, and maybe some magnetic field energy that biases it as well. And in this model, Ising model, the energy is really a physical thing. In the Hopfield model, in the Boltzmann machine, the energy is a mathematical metaphor. It gives you some value because there's all these tools associated with it. And that makes systems more tractable. But there isn't an actual system that has that energy. OK. So I think where I'm going with this is that we're going to take the Boltzmann machine model and ask, well, what is a physical system for which this actually is the energy? Then we have a system that has all the mathematical properties of a Boltzmann machine in the sense that it can represent probability distributions, but its behavior is actually just what physics does. The second point I'd like to make here is that, in some sense, when we look at things like the Ising model, behaviors like magnetism are sort of things that happen when you have many, many copies of the same thing. All the connections between units are identical. It's just J. But when you sort of say that there are more kinds of things, every neuron is actually different from every other neuron. Every weight between neuron I and neuron J is a different weight. You sort of have this more kinds rather than just more. And that brings you to a new place. So where this sort of interfaced with my work, I was very interested when I was learning molecular biology in how, as previous researchers like Erik Winfree noticed, genetic regulatory networks formally, mathematically, look very much like continuous Hopfield networks. They can have attractor states. You can write them down in very similar form. And so one of the first things I did when I was an assistant professor with my student Jungmin Kim was to sort of take a boiled down, simplified model of genetic regulatory networks that you could sort of create synthetically in a test tube with no biology, but using enzymes to produce RNA whose concentration level was analogous to how much a neuron is firing, its sort of firing rate, and whose interactions with the DNA templates, this RNA would basically switch on and off templates due to some clever designs. And that would be analogous to essentially a weighted sum being used to determine sort of, as a function of how much RNA there is, how much different genes would be on or off, changing those levels. But this is very much a non-equilibrium system. You're synthesizing new RNA, you're degrading RNA, you're going to actual point attractors in the way that we developed it. And so energy was, again, just a metaphor, more like a Lyapunov function. Later, with my student William Poole, we realized that you can talk about a set of chemical reaction networks that, in the stochastic regime, actually have an energy that's a physical energy, and that their stochasticity is analogous to the behavior of a Boltzmann machine. And these systems can do inference in the same way that Boltzmann machines do. Later, sort of the same analogous kinds of phenomena came up in studying molecular self-assembly, where the energy related to how well molecules stick to each other, again, defines an energy landscape that allows the system to do computation. And then, most recently, and what I want to tell you about, is a model of multi-component liquids where the phase separation process that allows the molecules to stick to each other and form clumps, again, has an energy that is in correspondence to Boltzmann machines. Okay, so I'm just going to very quickly give you just the flavor of William Poole's work. So the idea is you have some real cell, artificial cell that we're representing as having a set of chemical reactions that have detailed balance rates. And so you might have, for example, in this reaction, species A0 and, species B1 is catalytically converting the gray molecules to the black molecules. So in general, if you have a stochastic chemical reaction network, you have some species, they have some counts, and they have some energies. You can write down the full free energy of the system if you have a certain set of counts, and that's related to how many molecules do you have with their energy and an entropy term. And you get a Boltzmann distribution at equilibrium in that sort of well-mixed system with the molecules coming off each other, bouncing off each other. What we were able to show is that if you give me a Boltzmann machine that has some interesting distribution, MNIST, whatever it is you want it to be, maybe something with more relevance to a cellular environment, William can write down a set of chemical reaction networks that are detailed balanced and that have the exact same distribution. Furthermore, you can work through the same math that Hinton used to show that gradient descent on the relative entropy gives you a rule for changing the energies of these species with a contrastive wake-sleep type form, where now rather than looking at correlations, you just look at the counts of the molecules, and you look at the average counts. This is only interesting if your chemical reaction network has some very special structure, but you can treat arbitrary detailed balanced chemical reaction networks in this sense. Here's an analogous thing with self-assembly. So this insight was Arvind Murugan's with Stan Leibler when he was a postdoc. He was interested in jigsaw puzzle pieces that can come together in multiple ways. So the same pieces can fit together this way, which we'll call red, this way that we call green, this way that we call orange. And there's an energy landscape for, there's essentially an energy for a given partial assembly. So as you start putting things together, if we think about the ways these pieces fit, you can have an energy that says sort of how much do these sort of piece alpha and beta fit together if they're arranged as in a particular one of these shapes, and that's some energy that you can define. There's a chemical potential, and then you get the energy of a particular partial assembly as being this quadratic form with the variables x that say if tile alpha is present, it's a 1, otherwise it's a 0. And so you have this thing that looks very much like a Hopfield model. And in fact, I think more interestingly, if you ask, well, if you're this shape, will a particular one of these other pieces be able to stick into it? That's a step where something comes in, binds on its neighbors, and it attaches favorably if this linear weighted sum is greater than 0. Exactly the rule that you see in neural networks, except now this is talking about an assembly process. He was able to show further that this kind of process allows you to do a kind of associative pattern recognition. I'm going to skip this and jump to the point that when we got together, we recruited an alumnus from my lab, Konstantin Evans, to actually demonstrate this experimentally. So Konstantin designed 917 molecules, DNA molecules, that have different domains so that they can stick to each other, so that you can design some shapes that H, A, and M, such that the nucleation pathway, depending upon a pattern of concentrations, so these are images, but they correspond to a pattern of concentrations of tiles. Depending upon different patterns of concentrations, you will get different nuclei that will trigger the growth of different shapes. This is analogous to an energy landscape where, depending upon sort of where you start, you will have a barrier, a small or large barrier to rolling down into this basin or that basin or the other basin. And that process of nucleation is essentially recognizing, classifying different concentration patterns into different basins of the assembly process. So these are the actual shapes that we designed using these DNA molecules that self-assemble. Okay. So that was just trying to say hot field associative memory is something that can happen in molecular self-assembly, where the energy landscape is sort of has the properties that we're looking for. So now I'm finally coming to what I want to talk about, which we call Boltzmann liquids, which is a model that's self-assembly, but a liquid self-assembly rather than a sort of crystalline self-assembly process. This is theoretical work, but it's sort of motivated by a long sort of history of being able to design molecules that have programmable interactions in the liquid phase. So these are called DNA nanostars. They have little sticky ends of DNA that can stick to each other, and depending upon how similar these sequences are, they will stick with a different delta G. So you can program different nanostars. In this case, pink sticks to pink, and orange sticks to orange, and they'll phase separate into these little droplets. But you can, in general, design 900 of these if you wish, and design the sequences at the ends so that have a very particular pattern, Gij, of interactions between them. And that's what we're doing in the laboratory now, but I don't have anything to show you. So what I want to sort of show with this illustration is that a model of these liquids, programmable liquids, again, can be interpreted with respect to Boltzmann machines. So if you ask, what's happening inside the cell, things are incredibly dense. They're almost always sticking to each other. There's almost always some kind of energy between nearby molecules. Sometimes that leads to phase separation. Sometimes it doesn't. But if we think about having a bunch of species that have different stickinesses between them, that can allow us to study sort of the complexities that are inherent in phase separation. So basically what I'm going to say is that if we have a model of sort of a lattice model of liquids that phase separate, there's an analogy to Boltzmann machines. So here is our model. You have a cube with lots of smaller cubes at every position, and the energy of a particular arrangement of where all the molecules are is just a sum over every position of the stickiness of the molecule type that's at position X and the molecule type that's at neighboring position Y. So those are the molecular interactions. And then there's a free energy if we are working in a grand canonical mode where molecules can come out into and out of the box. If you're working in a canonical mode, then molecules just swap around inside the box. Okay, so if you have some interaction energies, this is the energy matrix for this particular simulation. You can see that some kind of phase separation happens. And again, based on this energy, there's a Boltzmann distribution of states of the box. Okay, so in what sense do these do inference? How do you clamp things? Well, if you don't update some of the units, for example, there's a little patch on the side, you can have a surface clamp. That's sort of like an external something outside of the part that we're paying attention to, the environment, if you will, holds a set of molecules in one place. And then the molecules that are free to arrive will do something onto that surface. And so what we see here is that this sort of clamping the identities of what's on the surface will give us a distribution of where all the other molecules go. You might just clamp a line. So here you can see we chose to clamp it with a sort of avocado pattern. And again, the conditional probability, given that these are what they are, is that there's going to be a droplet, an avocado droplet that localizes there.
So Erik?
Yes.
So biochemically, that would amount to fixing, almost gluing certain polymers at certain positions within your system?
Yes. So a kind of experiment that one can do is you have a glass slide, and you can localize some little DNA molecules in a particular pattern on that glass slide. They're fixed. And then you ask, as the solution of free monomers floats around, will they stick to that location and recognize it, or will they not stick to it and just keep on floating somewhere else?
So it's not just that you form those particular DNA, you've actually fixed them in space location.
You can imagine that, but it's a very general concept. For example, this concept is actually motivated by imagining this happens in genetic regulation. A linear polymer that has a pattern of binding sites, and you have molecules that stick more or less to that pattern of binding sites, and they can recruit a condensate of many different proteins in a liquid phase. This happens. Our point here is that that process of recruiting a droplet of or a condensate of proteins is performing a neural network-like computation to recognize that particular pattern.
Sorry, so you said the parameters of these systems. What counts as a parameter in this system?
I'll get there in just 1 minute.
Gotcha.
Parameters can be learned. So this is the task. We want to, rather than just say, here's an example of a weight matrix, see how it behaves, we say, I would like to teach a system to make these avocado-like droplets. These dotted bars are just sort of allowing us to see inside. So that's our training set. The parameters that we're going to change are the...
Just elaborate that. That's a singleton. That's avocado-like droplets. Are you...
So
Maybe there are other kinds of droplets as well? What's the multiplicity in the training set?
I'll show you some other examples. So in this case, this is sort of a dumb thing that one can do, which is try to train a network on a point. But really what that corresponds to is any shift in the space. And we could have wiggled the shape a little bit if that's what we wanted. We wanted a wiggly droplet. What we're seeing is that when we try to train on this point distribution, we actually get a much broader distribution because our model can't represent that point distribution. But it does what it does, and it does give us avocados in this case. So the parameters that we have are the Gij, which is how much species type I sticks to species type J. And again, we do gradient descent on the relative entropy. And it turns out that in this case, the sort of critical thing to know is in a given state, you can count how many interfaces are there between type I next to type J. So how many blues are next to yellows. And we have, again, this contrastive wake-sleep type behavior where we increase the energy when it's running free by looking at the average, how often is yellow next to red. And then we decrease the energy, making it more favorable when we clamp it to our target distribution that we're trying to teach it. And so that works in this avocado case, but is maybe not as exciting. And so to see sort of the exciting, we want to see some features of neural networks sort of come into play in the distributions that are represented by these Boltzmann liquids. So one way of thinking about it is that generalization is a phenomenon that happens, but that's also related to, well, what kinds of probability distributions can this model represent? If you try to teach it something that it can't represent, it won't do so well. So we don't have any theorems about that, but we've been exploring it so far with examples. So we might want to ask, is there something analogous to a Hopfield network where you have different sort of patterns that are known? So an image corresponds to a particular subset of now not just 4 species, but many species. And an inference might be if you clamp some of those species, will it recruit the other species? So the way this sort of comes into play for condensates is that our training set will be droplets that contain some mixture of species and other droplets that contain a different mixture of species. So in this example, this droplet has these colored species present. For example, has species 2, species 4, species 5, species 8, et cetera, including species 16. That's one memory of a composition. This is another kind of droplet we want to make. It has a different composition that shares some species. So if we do the wake-sleep learning on this is our sort of training set, we get a set of parameters. In this case, these are the Gij energies between species I and species J, how much they want to stick to each other. And this is a grand canonical simulation, so we also have some free energies that aren't shown. 5 minutes. OK.
So it's a quick question here. Why do you use wake-sleep rather than simulate annealing, which would seem to me to be more natural thing to consider, just reducing the temperature?
When we run it, it's just simulating at a constant temperature. So we actually use simulated annealing in another example I talk about, but that's just trying to get to equilibrium because we're not there yet for looking at more sophisticated things. But the wake-sleep is for training the parameters. So simulated annealing per se doesn't tell you how you can change the energy between, say, red next to blue. OK, but once we have established a set of weights between the different molecules, how much they want to stick to each other, we can then clamp on the surface part of composition 1, and it recruits the rest of composition 1. We can clamp the surface with composition 2, and it recruits the rest of composition 2. And so this is an example of this equilibrium process doing inference in a multi-component liquid. It's interesting to argue, I'm sure there are many subtleties I'm not aware of here, but the way I think about it is that the environment is providing energy by clamping the surface, but the stuff going on inside the box, what the cell is responsible to, is just in equilibrium rearranging itself in response to the energy provided by the environment. And so this is a energetically free, very efficient way of doing inference. OK. I'm going to skip a few things and wrap up. This is just one example that shows that if we can do sort of the Homer Simpson example, if we clamp not to the full state, but just say these 3 species, here's our composition. We run the system, we let it go to equilibrium, and look at how many molecules we have of different types. Apologies for the color map here, but this is saying that it recruited quite a bit of species 1, it recruited quite a bit of species 4, it recruited quite a bit of species 6, et cetera. It basically recalled composition 2 because we clamped to a subset of composition 2. So this is doing Hopfield-like associative memory recall. OK. There's an example that has hidden units as well. I think I will mention this. So these Hopfield droplets were looking at, basically we trained them on a distribution so that we wanted them to make droplets, but we wanted the composition to be a particular mixture. It was either this mixture or that mixture. Those were the 2 memories. But each droplet was well mixed in the sense that there wasn't any spatial organization of the molecules other than being in the droplet. We wanted to ask, since this is a model that has a spatial dimension, can we use wake-sleep training to find energies for a multi-component liquid that can recognize spatial patterns? So what we wanted is some molecules where one set of molecules will basically stick to a checkerboard pattern, whereas another set of molecules will stick to a stripe pattern. And since Wake's, basically Hinton's classical Boltzmann machine model can deal with hidden units. It's the same learning rule. We thought that might work here as well. And it did. So our training examples were things where we asked it to recruit red on near the checkerboard and blue near the stripes, and we let it run free for sort of the molecules that are gluing them together. And it came up with a weight matrix that does, once you let the system just run completely free, recruit red to the correct side. So now we have a spatial pattern recognition. We explored another variant where we essentially, more like the Hopfield networks with the counts of species within a membrane, now with some species being permeable in and out of the membrane and others not, but still with a well-defined equilibrium mixture. We showed that you can, it can learn an MNIST distribution, sort of, not so great, but a little bit. So although we don't have a sort of universal approximation theorem for what kinds of distributions Boltzmann liquids can represent, we do have some of these examples of interesting enough distributions. So I think I can now wrap up and try to argue that if you're trying to look for sort of where the information processing is happening inside some molecular system, biology, of course, has worked very hard to get the most mileage out of the components, get the most work done out of a very compact space with its given resources. And so we might expect that some principles that are sort of neural computing principles arise naturally from the energetics of equilibrium subsystems inside the cell, or perhaps inside of artificial molecular machines that we might design in the future. Thank you.